Good evening, everybody, and welcome to the first talk in the Keele IOP Physics Centre lecture series for 2021-2022. I think we had about 70 or so people sign up this evening, so I'm hoping, I can't see, but I'm hoping there are as many of you there this evening. So just to give you a few introductions to those that maybe haven't attended one of our talks before, currently we are delivering all our talks virtually, as we are doing tonight. There is a hope that we can move back towards in-person or a hybrid model in the new year. And I will, of course, keep everybody up to date with that. You may see in the chat box, I've left the link for our mailing list sign up. So for anybody that would like to find out more about our future talks within the series, please do complete that very simple MS form, and I will ensure you get all the latest updates. So, in terms of the way this will run tonight, we will have our speaker, who I'll introduce very shortly, who will give a 50 to 60-minute talk. Around halfway through, we'll take a short break to allow Stephen, our British Sign Language interpreter, a short break, and as well as ourselves to fill our glasses or nip to the toilet. For those that might require it, we have moved from GoToWebinar onto Zoom, which does allow us the closed caption option. So for those that require that for accessibility, please do make use of it. You can find that on the scrolling toolbar at the bottom of the window. So with no further ado, I'm going to introduce tonight's speaker. And in so doing, hopefully we'll kick off our wonderful talk series. So our speaker for this evening is Ard Louis, who is a professor of theoretical physics at the University of Oxford, where he leads an interdisciplinary group of researchers. His recent interests include the physics of biological evolution, self-assembly, and theories of why deep learning works so well. He was born in the Netherlands, but grew up in Gabon in Central Africa. He undertook his first degree in Utrecht and his PhD in solid-state physics at Cornell University. Before coming to Oxford, he was the Royal Society University Research Fellow at the University of Cambridge. He's also, interestingly, recently made a 4-part documentary called Why Are We Here with David Malone, and appeared in The Story of God with Morgan Freeman, giving him an Erdos-Bacon number of 6. Outside of science, or perhaps inclusive of science, maybe we'll find out some of this, his hobbies include molecular gastronomy and wine tasting. So with no further ado, firstly, welcome to everybody that's joined us this evening, and I will hand over to Professor Ard Louis. Thank you, Ard.
Well, thank you very much for the invitation. It's a great honor and pleasure for me to speak to you here. I'm speaking to you from my kitchen. I'm having a little glass of wine. I hope some of you are able to enjoy yourselves too. One of the funny things about this virtual world is that we're watching one another and seeing one another in all these different, in our own home environments. And so although I would prefer to be there in person, I thought I'd take advantage of the fact that, that I'm at least at home. So what I'm hoping to convince you today is that there are some really interesting fundamental theories that will predict in some way or the other that the world that we see around us is simple. Now, we often talk about Occam's razor. We think the world is simple in some way. Descriptions are simple. Physicists have often said this in many ways. Einstein, Dirac, many of the greats have talked about the importance of simplicity, the importance of elegance. And I'm going to try to convince you, hopefully, that there is a theory that I think every physicist ought to know about that actually predicts this quantitatively. But before I do that, I want to start with a story. And it's not really my story. The story is that by a very famous Argentine writer, Borges, one of the kind of the very early magic realism. And one of his many great books is actually more of an essay called The Library of Babel. And it's a library where every book has exactly the same number of pages, 410 pages, 4 lines, 80 characters. It's an alphabet of 22 letters, along with period, space, and comma, so 25 characters. And the interesting thing about this library is every possible set of characters is in there. So every possible book of length of this length with this particular set of letters. And so in the story, people go into the library because in that library, there is a book that gives you exactly your entire life story. And not only that, but it'll tell you what's going to happen to you in the future. So people go in there looking for their vindication, the story about the future. But as Borges points out, it's dangerous because there can be all kinds of treacherous variations. So in that library, there exists a book that tells the entire history of Ard Louis. And in 5 seconds from now, an asteroid comes through my kitchen here in Oxford and blows me to smithereens, for example. That book is also there. Any book you can describe in 410 pages is there. So although every book is equally likely, and there are an extraordinarily large number of the book, there's basically 10 to the 1.8 million possible books. Every book is likely, but not every story is equally likely, right? So maybe my life has been very complex, and so the book of my life will be very complex. But maybe there are people with simple lives, or maybe there's a book that tells us a very simple story. And if a story is simple, you might imagine that it could be told in many different ways. And so you might find that if you coarse grain a bit to the level of stories, that every story may not be equally likely. If you like these things, there's a Library of Babel.info. You can type in anything you want, and it'll tell you where this is found in any book, because every single combination of characters is there. So that's the kind of thing I want to talk about. And another kind of intuition I want to remind you of is how false exponential growth is. So in the great Persian epic poem, Shamana, on the Book of Kings by Ferdowsi, he retells a story of the inventor of chess who comes to the king after he's invented chess. And the king thinks this is amazing and wants to show his grandeur by rewarding this inventor with some great gift. And he says, ask me anything you want and I'll give it to you. And the inventor says, well, I would like to have one barley corn on the first square, 2 on the second, 4 on the third, and just doubling every single square. And at first the king scoffs because he thinks he's unhappy because his great generosity hasn't really been properly acknowledged, until his accountants start adding it up. And it turns out that if you do this, you'll actually have 18 quintillion grains of barley. If you were to double barley corn on every single square of a chessboard, you would have roughly 10 times the world's current grain production that you'd have to produce. And so this is actually one of the 64 doubling was a very famous problem on the Silk Route. And my children are doing Silk Route right now in school. And so it was a problem. It's actually 30 doublings and 64 doublings were two kind of amusing things to do. But it just shows you how rapidly doublings or exponential growth is. And so I'm going to tell you 3 stories today that are all in these very large spaces. In fact, I call these hyper-astronomical spaces because they're spaces that are much larger than you, and you quickly reach for kind of astronomical metaphors. So the first one I'm going to tell you about is evolution of RNA. So I just told you that doubling problem. So in our bodies, we have a number of really important molecules. One is proteins, which is the kind of molecular workhorses of the cell. These are linear sequences of an alphabet of 20 amino acids. And we have RNA and DNA, which are molecules which can store information. RNA can also act catalytically like proteins do. So it has a dual role, which is why we think it might have been involved in the origin of life. And it has an alphabet of 4 nucleotides. So you might ask yourself, well, how big are these spaces that nature has to search in? For proteins, interestingly, if you made every length 58 peptide, that's relatively short for proteins. The mean length of proteins in your body is 476. It would weigh, if you made every single one, it would take you more matter than the matter of the observable universe. That's why we get to these hyper-astronomical numbers larger than the universe. And for RNA, which is a system I'm going to study today, it's 126. So if you have a length 126 RNA, which is a relatively short RNA, if you made every one of that length, it would weigh more than the observable universe. So these are very, very, very large numbers. The mean length of proteins in your body is 476. If you made every protein of length 476, it'd be 10 to the 500 times the mass of the observable universe. So this tells you these spaces are very, very big. And given that space is very big, I want to tell you a very surprising story. So Jack Szostak, a Nobel Prize winner at Harvard, did an experiment about 20 years ago in his lab. He does experimental evolution. So that basically means in the lab, you have a bunch of molecules, in this case, RNA, random RNA molecules, in his case, length about 55. You mutate them, and then you select on some assay. In this case, the selection was on self-cleaving enzymes. So an enzyme is something which catalyzes a reaction. And so it's a particular enzyme. For physicists, you don't need to bother what it is. It's just a particular function. This thing does something chemical. And so you fish through all the molecules, look which ones have a little bit of that activity. You pick out the best ones, and then you mutate those again. And you do that again. You repeat until you get a high yielding a high quality enzyme. And this is a very interesting type of set of experiments. In fact, it works remarkably well. So he did this, and what was really surprising to me, is every time he did the experiment, he found not just a self-cleaving enzyme, but he found the same shape, which is called the hammerhead ribozyme. It has this kind of shape. So this basically, these things fold into these three-dimensional shapes. This is the one that it folds into. And it's interesting, the hammerhead ribosome is also found in nature. It's found in animals, plants, bacteria. It's found everywhere in nature. So the question is, was it obvious you should find that every time the same thing? Well, if you made every length 55 RNA, so the size of the space of all possible RNAs about 55, that weighs about 10 to the 10 kilos, 4 to the power 55. I estimate the experiment used probably at most 1/100th of a gram of material. And so that's only 1 in 10 to 15th of the space. So why this evolutionary convergence? Why does this evolutionary process converge on the same shape every time? It's a little bit like taking a haystack, putting your hand in and finding a pin. And then you put your hand in again, and you find the pin again. And every time you put your hand in, you find the same pin. That tells you something funny is up. Right? So this experiment actually was what got me interested in thinking about how does this work? The space is unsearchably large, and yet you find the same thing every time. What's the trick? Well, one of the very nice things about RNA is that it's a relatively simple molecule to do certain kinds of calculations on. So here I show you a sequence. So I told you I've got 4 letters. So they've got G, A, U, and C are the 4 letters. The first order, G binds to C and A binds to U, although it can be a bit more complicated than that. And so what you have is I have this linear sequence here of letters of length 55. And then I can ask you, what is the lowest free energy state that these things will fold into? For example, what if I look at all possible ways they could fold, what's the lowest free energy? And while I can't calculate the 3-dimensional structure, that's a very hard thing to calculate, it turns out I can calculate quite quickly a simpler thing, which is basically just who binds to whom. Right? So in this case, this particular hammerhead ribozyme is equivalent to this particular what's called secondary structure. And it basically says, for this sequence, the 2 outer G's don't bind to anything, then this A here binds to that U there, etc. And as you walk down, you can kind of walk through all the things that bind and don't bind. And the ones that are next to each other are bound to each other, and the ones that are loops are unbound. So that secondary structure problem is quite important, because once I know the secondary structure, then I can more or less predict what the 3D structure is. That's in fact what Shostak and his collaborators did, is they measured that they looked at the sequences, they calculated the secondary structure, and they found that it gave you every time this characteristic hammerhead ribozyme structure. And they didn't seem to ask themselves the question about how is this possible. It's a classic kind of biologist, of course it's possible because it happens. But physicists like to ask themselves, why? Why? How does that work? And so I started working on this, and we started doing these calculations. Now, these relatively large lengths are hard to calculate exhaustively, but you can do shorter ones. So here is a length 15 RNA. I'm just going to walk you through how this works. So here is the ground state free energy shape for this particular sequence, which starts G, C, G, G, right? So G, C, G, G, A, A, U, it goes around like this. And these green lines are the bases that bond, and these ones are the ones that don't bind. So G binds to C, I told you C binds to U. Actually, G sometimes binds to U, so it's a little bit more tricky, but G binds to C. So what's interesting is I can look at this shape. I can abstract out a little bit. I'm going to abstract out into a representation that I call dot bracket representation. This is traditionally used by biologists that look at this. And what it basically says is, every time I have a bond, I make a bracket. So open bracket, open bracket, open bracket, open bracket. Every time I have a no bond, I do a dot, dots, 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 dot. Close bond, close bond, close bond. So close bracket, close bracket, close bracket, dot, dot, dot. So you see here, bracket, 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 open, dot, dot, dot. Those are the 4 here that are unbound. Close, 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 dot, dot, dot. And these two are exactly this equivalent to each other. So what I can ask, this is the following question. If I give you this particular shape with these brackets, how many sequences have this shape as its ground state? Turns out you can calculate that for relatively short sets of sequences. And so we did this for all length 15 sequences. There's 4 to the power 15, which is 2 to the 30. Just remember that 2 to the 10 is 1,000, 1,024. So 2 to the 30 is 1,000 cubes, which is basically a billion. So there's a billion sequences coming down on a computer relatively quickly. So we folded all billion sequences, and what we found is that although there's a billion sequences, there are only 431 shapes. So already this is telling us something about Shostak's issue, which is although the space is very large in terms of sequences, the number of things that they actually do is less. And that's helpful because that at least gives me a higher probability that I'm going to get something. I just told you this story, then you say, well, okay, for length 15, rather than a 1 in a billion chance that I get the same thing twice, there's a 1 in 431 chance that I get the same thing twice. But I can go further than that. I can count how many sequences map to each substructure. So how many sequences have that particular structure as their ground state and or the lowest free energy state. And so this one has 11,000, almost 12,000 sequences mapping to it. This one has only 2. So there's only 2 sequences for which this structure is the lowest free energy structure. And so if I take the number of sequences that map to a structure and I rank them from 1 to 431, I see this very steep decline. So there's a small number of structures that have lots of sequence mapping to them and lots that have almost nothing mapping to them. In fact, only 26 of the 431 shapes take up 50% of this space of all possible sequences. And this red line here is basically telling you everything above here, it takes up 50% of the sequence space. So if I just grab a sequence at random, which is more or less how Shostak starts, I've got a 1 in 26 chance, 50% chance, sorry, 50% chance to get just one of these 26 ones. So these are quite frequent. So I might expect them to appear with high frequency. So this is a little bit like, these are the needles that I'm going to find quite frequently when I randomly grab into that big haystack of sequences. So that's kind of nice and interesting. That's giving me already some hint that perhaps what Shostak is seeing is a very strong bias in the mapping from sequences to structures, such that when he randomly shuffles his sequences, with very high probability, he gets a small subset of structures. Now, what we've done since then is worked out some mathematical techniques to calculate how many sequences map to a structure for arbitrary lengths. So for length 55, there's 10 to the 33 sequences. There's no way you can do that exhaustively. We estimate there's about 10 to the 13 possible structures. Okay. And here on this plot, this black line tells me the neutral set size, which is a bit like the entropy or the number of sequences mapping to a structure. How many sequences map to a structure? So an entropic argument. And so the large entropy structures that have many sequences mapping to them, or the one here way on the left, some of them have 10 to 25 sequences mapping to them or more, very large numbers. These over here are the rare ones, 10 to 13 of them in total. And then what we did, we found something extremely surprising to us, but also quite exciting. There's databases that biologists have put together of what are called functional RNAs. So just to remind you very briefly, RNA in the cell gets copied as messenger RNA from DNA to the ribosome to make proteins. So that's basically just a copying of information from DNA to RNA. But there's RNAs that are functional, so they're not information carriers. They're doing something like catalysis or structure. Those are the ones we're interested in. And people have put these into databases, curated them, and when we looked at the last time we looked at the database, there were 504 RNA sequences found in nature of length 55. And what we did is for each of those sequences, we mapped what the structure was. And then we noticed that they're all over here on the left. Okay, so they're all the structures found in nature are those structures that are very likely to be found on random sampling. And that hammerhead ribozyme is one of them. So we think that this probably explains why Shostak and his collaborators kept finding the same structure. It's a structure that's just really easy to find. Okay, so it's that might be a tautology, but we show you why it's really easy to find. It's really easy to find because it's one of the structures that has many sequences mapping to it. Interestingly, there are 10 to the 13 possible structures. The ones that are actually found in nature are a tiny, tiny, tiny subset of that. So nature has only explored an unbelievably tiny subset of the kind of shape space that's in principle possible. And that's a kind of interesting little point to think about. And so we've actually generalized that out. So if you look at these structures, this is more of a technical thing. So here's a complicated structure. This is called a glycine riboswitch, if you like these kind of things, but don't worry about it if you're physicists. It's actually an RNA that switches between 2 different conformations. This is a secondary structure. And so if I find, a genome of some organism, I look at the non-coding or the junk DNA bit, and I find a sequence that when I fold it looks a bit like this, and I'll think, that's probably a glycine riboswitch. Now, if I look at that, you can kind of think to yourself, well, is it really going to be that different if this loop has one more or one less base in it? Probably not. If the stem is a little bit longer, a little bit shorter, probably not. And so people have said, well, let's look at these in a coarse-grained fashion. Let's, instead of looking at the full dot bracket notation, let's simplify this to looking at a few simple coarse-grained aspects of it. And this is a more of a bioinformatics detail, but you can work this out at various levels, and you're basically coarse grading over structures. So lots of structures that are kind of similar to each other will all fall into the same category. And the reason this is interesting is because in nature, there's 500 length 55 RNAs, but there's 10 to 13 structures. So the probability that you find the same structure more than once is relatively small. But by grouping them together in these coarse-grained shapes, I might get enough ones found in nature that I can actually start seeing how frequently a particular shape appears, a coarse-grained shape appears in nature. And so here, we've done this for length 55 RNA and length 126, which is the one for which the total number of sequences would weigh more than the mass of the observable universe. And we find three really surprising things. First of all, that the frequency, so how likely they are to be found randomly, is very highly biased. Okay, so here there are only about 28 out of 10 million possible structures are found in nature, and the most likely ones to be found are the ones here at the top, and the natural ones are exactly those, with a few exceptions down here. So out of the 10 million structures that nature could use, 28 are used actually in practice. And those are also the ones that we predict would be used by just random sampling sequences. So those are the ones with the largest sequence entropy, the most likely to appear. And the same is true here for length 126 RNA, of which there are an hyper-astronomically large number of possible sequences. And we think there's about 1 trillion possible structures at this particular coarse grain level, and only 25 are found in nature. And those 25 are the most likely ones to be found. So that's very surprising, because that tells us that if I just randomly pick sequences, in fact, in this case, I only picked about 10 to the 4, maybe 10 to the 5 sequences, okay, 10 to the 4 sequences, 10,000, 100,000 sequences. I actually find all the structure at this level that nature uses, all the structure that's needed for life in all its glory to work on our planet can be found in this relatively small fraction of the space. So this is kind of interesting, because I just told you the space is hyper-astronomically large, and it is, but nature's only used a very tiny fraction of that space. And I've just shown that pretty clearly here. What's even more that's kind of surprising, right? So this is a surprising thing. It's a lot more that we've written a paper about this. It's just come out actually, but this is the bioarchive. You can read it, where we describe this and a whole bunch of other interesting things. In fact, rather surprisingly, these highly frequent ones are also much more, they not only appear, they're not only the high frequency ones, but they also appear more frequent in nature. So we can even predict how often you'll find certain RNA shapes in nature, not only which ones you find. And so that's all quite surprising, because I've ignored natural selection entirely. Natural selection is a normal big story of evolution, and rightly so. But here I've explained something quite fundamental about how the natural world works, how RNA works. And I've not had to use natural selection at all. I've ignored natural selection entirely. Natural selection is still working, obviously. So in the case of Shostak experiments, natural selection is what gives them that particular enzymatic activity. But why do you have this particular hand-made ribozyme? Well, it may very well be that is simply caused by this entropic argument. And that there are, in the 10 to the 7 other possible structures at this level, many other structures that will also give you the self-cleaving enzymatic activity, but they're simply never found in nature. So the big question then is, why do we have this incredibly strong bias towards a very small subset of this very big space? Well, there I want to tell you a story about information theory. So in physics, you're used to Shannon information, entropy, which is really Shannon information. So it's like a measure of surprise. It's something about distributions. But in the 1960s, a number of very brilliant mathematicians started thinking about a different question, which is, what is the complexity of a single string? So let's say I give you a string like this one, 0, 1, 0, 1, 0, 1, repeated 50 times. Well, intuitively, that's a simple string, right? That's a string that has not a lot of information needed to generate it. Whereas if I have a complicated string like this, which is just, as far as I know, a random sequence of zeros and ones, it's probably quite complex. And what Kolmogorov and Chaitin, two great mathematicians, worked out is the formally correct way of distinguishing those two sequences is by something called Kolmogorov complexity, which is the length of the shortest code that describes that particular sequence on a universal Turing machine, which is a computing device that can do any calculation. So it's kind of interesting because this tells us that it kind of picks up the intuition that this first sequence could be described by print 0, 1, 50 times, which is a short description, whereas this long one probably has no description otherwise other than print itself. And in fact, it turns out that the best, a formal definition of randomness, what's a random sequence, is a sequence for which there is no shorter code than print the sequence. So therefore, because you have to add the print statement, it's a sequence for which this Kolmogorov complexity is longer than the sequence itself. Another kind of interesting and strange intuition you pick up from this is that the complexity of a set can be much less than the complexity of the elements of the set. Think of Borges's library. I can describe that to you in a relatively short amount of space. So the complexity of describing the entire library is relatively short. All books of length 410 pages, blah, blah, blah. However, in that library are books that describe your life and very complicated things. There are books that describe general relativity and great, and there's probably a book there that has solved, there is a book there that explains exactly how you combine quantum mechanics with gravity. So there are very complex things in that library, but the description of the library is simple. So the complexity of a set is much less than the elements of the set. This is kind of a fun thing, and we're going to use that in a second. And then how am I going to use that? Well, I'm going to give you an intuition about monkeys typing on keyboards, and you'll see in a minute why this Kolmogorov complexity comes back. So how likely is a monkey to type out the first m digits of the number pi on an n-key typewriter? So let's say my typewriter, my laptop, I say now because not everybody knows what typewriters are anymore, the young people don't. I randomly type, if I've got 50 keys, I got a 1 in 50 chance of getting the correct digit at each time. I need to, if I want to have it length m, I need m plus 1 because I need to get the dot there, 3.141592. That's how likely I am to make pi. But actually, it's how likely I am to make any sequence of that length. They're all equally likely or unlikely. So that's the probability of monkey randomly typing into a typewriter. But what if I twist this trope and ask myself, what if the monkey types into the computer program C, for example? How likely is the monkey to generate pi? Well, this turns out to be a branch of recreational computer science, or there's even competitions on it, write the shortest program that will generate pi. And here's what as far as I know, the shortest code I know of by Dick Winter in Amsterdam, where this 133 character code in C will generate the first 15,000 digits of pi using something called the spigot algorithm. And so that's kind of fascinating. If the monkey's typing not into a computer or to a word processor, but typing into a computer program, they might accidentally type with 1 over n to the power of 133 this program. And if they do that, they're going to generate 15,000 digits of pi. So in other words, if I look at the outputs of a monkey typing into a computer program, what I'm going to see is that outputs that have short descriptions are going to be exponentially more likely to appear than outputs that do not have short descriptions. And that's a really interesting idea that can be formalized. And it was formalized originally by a man called Ray Solomanoff, who was asking exactly this question. If I have a computer and I randomly type code into it, just randomly type into it, how likely is it going to produce a certain output? So he simplified this to a computer which only takes binary inputs, so just zeros and ones, right? So I'm just randomly typing zeros and ones, and the computer reads these, and if it recognizes it as a code, it'll execute the code. Well, let's say I want to have that computer print some output, say pi, right? Then, well, how likely is that to happen? Well, it's going to be the sum over all programs that output pi, and then how likely is a program of a certain length to output pi? Well, it's 1/2 to the power of the length. So every program that will be produced with the probability 1/2 to the power of its length. What's the most likely program? What's the program with the shortest length, right? So that's 1/2 to the power of the Kolmogorov complexity. In fact, Solomon came up with this idea before Kolmogorov did, but of course, Kolmogorov is a great famous mathematician, so the name stuck with him. The great artificial intelligence researcher Marvin Minsky said not long before he died, it seems to me that the most important discovery since Gödel was the discovery by Chaitin, Solomonoff, and Kolmogorov of the concept called algorithmic probability. Everybody should learn all about that and spend the rest of their lives working on it. So why is this so interesting? Well, this is kind of a lower bound on the probability, right? Because there's at least one shortest code. There might be a very large number of longer codes, you're thinking. So why is this shorter code interesting? Well, a great computer scientist, Leonard Levin, also known for P and NP, worked out that actually the upper bounds is also given by 2 to the minus k. So the probability of you getting this particular output is bound to the lower bound by, there's at least one program that will generate. If this program will generate it, the shortest one will be that will be the shortest program. And if there are longer programs, they'll at most give you a kind of constant addition to the exponent. So intuitively, simpler outputs are much more likely to appear than complex outputs. This was proven back in the 1970s, almost 45 years ago, 43 years ago. You might wonder why you haven't learned about that in undergrads. And the reason is because there's some serious problems in applying this. One is that most problems that we're interested in aren't universal Turing machines. They can't do any calculation. Formally, the Kolmogorov complexity is uncomputable. It reduces to the halting problem, which Turing actually invented the universal Turing machine to look at Gödel's undecidability, which is a deep question in foundations of mathematics. And it only holds asymptotically for very large complexities. And so for that reason, it hasn't really been applied in practice that much. But we being physicists aren't going to let ourselves get too worried about the niceties of mathematical proofs. And so I was looking at this, I was actually trying to understand why the RNA maps were so biased. And I had a mathematics student who pointed this theory out to me. And then I said, well, okay, I can't prove that this is true rigorously for non-universal Turing machines, but this is a very strong result. I can't believe that it's going to just disappear entirely once I'm away from the regime of universal Turing machines. And in fact, there's a whole theory of computation, something called Chomsky hierarchy. I couldn't believe if you went down the Chomsky hierarchy, it would all suddenly disappear. And so we said, well, we're going to try something simpler. We're going to, for simple maps, we're going to take all the inputs, calculate all the outputs. And that is like calculating all the books in Borges' library that has short algorithmic information. Then we're going to order those books by the frequency which they appear. Then we're going to write an encoding for that tells you how you can most efficiently encode that. And if we use little tricks, then it turns out that we can actually calculate an upper bound on the probability which holds for any kind of input-output map. That's relatively simple. And so I'm saying it to you now in about 2 minutes. There's actually 2 PhDs of work by my students, Kamal Dingle, who's now a professor of mathematics, and Chico, who's now a professor of computer science. And so it's not non-trivial proof, but they did this. And it turns out that as long as the maps are relatively simple, that means that the complexity of the map does not grow quickly with size, then you can write down this kind of approximation that the probability that an output X appears is bounded by a half to the power of some approximation to its Kolmogorov complexity, to its how complicated it is. Well, I told you the Kolmogorov complexity is tells you something like the shortest code that you describe something. When you compress a file on your computer, for example, what is it doing? It's basically looking for some simple description of your file. Like if there's anything that repeats itself, it says, well, this word repeats X number of times, etc. It typically builds up a library of this, and that's how it compresses the file down. And so you can use compression probably as a kind of rough estimator of what the Kolmogorov complexity is. And so there's lots of technical aspects here, which you can read about in our papers. But the main point is that this is a prediction that we've made, and then we thought we'd try it out. This is a typical physicist approach, right? So we actually have lots of things that we can't rigorously prove the radius of convergence of, for example, but we use them nevertheless, and they work remarkably well. Even if we're outside of the radius convergence we can prove. That's effectively what's happening here. So here's an example back to RNA. This is length 55 RNA. Here I've given you for a bunch of different, these are basically different dot bracket notations in order of how likely they are to appear if you randomly sample sequences. So you see these quite simple structures have high probabilities or high log probabilities, whereas this one, which is relatively complex, is much less likely to appear. Several, in this case, about 5 orders of magnitude less likely to appear than this one is upon random sampling. And interestingly, I can then take these sequences, put them into a compressor. It's the same thing like when you zip on your computer effectively. We use something called Lempel Ziv 76, but it doesn't really matter which one you use to first order. This is a nice one for technical reasons. So what this does is this tells you that the more compressible a structure is, the more likely it is to appear upon random sampling of sequences, the larger the sequence entropy is going to be. And here we show for length 55 RNA, a whole bunch of randomly sampled RNAs. We show how often they appear. This one here is the simplest one, which is unbound entirely. And then here you have the other ones. And this black line is our bound with one constant fit. This dashed line is actually the bound with no fitting at all. It's basically just the raw prediction. And so we're able to predict really accurately how the probability and the complexity are bounded. Remember, it's a bound that we can predict. We can also actually interestingly predict something about the things that are far from the bound. This tells us a bunch of other things about them, but we can predict this. So to first order, simple structures can appear with high probability. High probability structures are always simple, and low probability ones and highly complex ones are always low probability. That's the right way of saying it. So this tells us something very interesting. The structures that are found in nature, this tiny subset of all possible structures that nature uses, the ones that happen to appear with high probability upon random sampling sequences, are also structures that are highly compressible. They're simple structures. So nature has a kind of inbuilt Occam's razor, an inbuilt preference for simplicity. And at least in this quite simple biological system of RNA, we're able to predict incredibly well what those structures are. And we did this for a whole bunch of other maps. I'll just talk to you about one or two. So here's a set of whole bunch of couple different equations, actually a model of an oscillator, a cellular circadian rhythm oscillator. And but you can think about that as a map of inputs to outputs. The inputs are the coefficients in your differential equation, on the differential equation. The outputs are the outputs of one of the variables. You can then look at the complexity. What you see is that simple patterns are much more likely to appear than complex patterns. And we can do this for a whole series of other systems. And every system that we've looked at, we can do that. And here's one which I won't tell you exactly how it works, but it's a matrix map for which one of our conditions doesn't hold. It's a map that grows with complexity, grows quadratically with its size. We predicted our theory should not work there, and indeed it doesn't. So it's always nice if you can break your theory in a predictable way. So this means that we can predict a whole bunch of things about these input-output maps. And I think at this point, it's a good point to take a little break. I think there's a 3-minute break tied in. Then I'll come back and just tell you 2 more short stories to finish off the talk.
Thank you, Ard. Yeah, so for those that would like to go and fill their glasses, please go and do so now. I'll pop to the loo. We'll just take 3 minutes and then we will return. If Matt Wayne is still there, Matt, I'm going to break our normal tradition. I'm going to allow you to ask that question that you've posted in the chat to Ard before we start the second part of the talk, because I think it'll be quite a nice timing. So when we come back for the second part, please have that ready if you could, and we'll pose that to the speaker shortly. Okay, I'll see you in a moment. Okay, welcome back, everybody. I hope that was a useful 3-minute interlude. Matt, so I'm going to break with tradition. I hope that's okay with you, Ard. I'm going to take a couple of questions at this point because I think it will nicely segue into the second and third parts of our talk. So Matt Wayne, Matt, if you are there and able to unmute yourself, you may not be able to. In fact, I've just been told by our director here that I may have to unmute you personally. So bear with me. I will see if I can do that, or if Daisy can hear me. If you could unmute Matt Wayne, that would be lovely.
Hello, can you hear me?
You can. Right, Matt, over to you. Could you just pose that question to Ard, because I think it's a really interesting one at this point.
Certainly. Going back to the, I was a couple of minutes late, so I missed the very beginning. But going back to the simplicity of the structures and how often they're found, do you think that's something which may be specifically true to life on Earth? So, for example, carbon-based life, or do you think that this may also be true about any planet capable of supporting life?
That's a very good question. In fact, I was just in Iceland recently for a workshop on, or partially on, origins of life. That was a question that was asked there. So, because people, when you're looking for life on other planets, should you look for kind of water, carbon-based life, or should you think there might be life based in silicon or something else? I think this argument is sufficiently general that it should hold for any kind of life that we see. I didn't make any assumptions about carbon-based life at all. It's just a property of the world. In fact, although I skimmed over very briefly at the end, I did give the example of the differential equations. And I'm just saying this property, this coding theorem, the idea that a lot of things we see in nature are processed. And so there's random typing of inputs, but they're processed. The inputs are not the outputs, but the inputs are processed. And as long as that's true with a few conditions, then you should see simple things appear with much higher probability than complex things.
Thank you. That's a very interesting answer.
Thank you, Martin. Thank you for that answer, Ard. Tariq and Penelope, I've noted your questions, but for the sake of brevity, what we'll do is we'll ask those to Ard at the end of the session now, so we can get through the talk. And if anybody else has any questions during that time, please post them in the chat or the Q&A, and I will pose them at the end. So back to you, Ard, for the second part of this talk. All
right. So the law. I'm going to tell you two more stories to finish this off. One of them is I'm going to show you this little video. There you go. This is actually a bacteria. Bacteria swim with these flagella on the back, like little tails that they spin around in the liquid which they swim. And those tails are spun by these incredible little motors. These are motors that can rotate at 100,000 RPM. They can stop in a quarter of a turn. Here in Oxford, I have a colleague, Richard Berry, who measures the torque of these machines and a whole bunch of other properties of them. Absolutely fascinating pieces of nanomachinery. What's interesting about them as well is that they're not made in factories like a motor would be, or any other complex machinery they're used to. They actually make themselves. So these things float around in the cell, and the proteins spontaneously self-assemble into these very specific, very well-defined shapes. And so this bacterial flagella motor, you're watching it now, an animation from Keiji Namba's group in Osaka of how this self-assembly might occur. It's an absolutely fascinating piece of science. In fact, I saw this about at least a decade ago, and I thought, well, how on earth can that be possible? So I've been studying self-assembly in my lab for quite a while since then. And the self-assembly is amazing enough in itself, but you can also ask yourself, well, how did we design something like that? So nature designs things by random mutations. How does it happen upon something that is that fantastic? It turns out that when you think about our bodies, they're made of proteins, which are the molecular workhorses of the cell. And about half of the proteins in our body are in fact found in some kind of complex. So famously, hemoglobin, which we need for oxygen in our blood, is a tetramer, 4 molecules stuck together. And these things self-assemble. So the hemoglobins are each made separately, and then they float around the cell and they come together in these tetramers. So you can study that self-assembly process. And here's a bunch of different protein assemblies you see here. And here's like a little graph of how their topology works. And we decided to simplify that to a typical physicist model of little squares on a lattice that have interactions that stick to each other. So this protein complex here becomes this little set of squares. These are called polyaminos, just like dominoes are 2, polyaminos are any number of squares. It's also a branch of recreational mathematics. It's quite fun to play with. And here's an example of one of those polyaminos. So here I've got a particular assembly mechanism, which I put one seed particle down, number 1, and then I let these other particles just randomly float in and out of the solution. And I ask myself, what will this thing make? Well, I've got here a set of rules, okay, and I've got a set of sides. In this case, A binds to C means a 1, and 0 means it doesn't bind to anything else. And so if 1 is down there, which has A's everywhere, then a particle, only particle that can stick is a particle that has C's on it. Well, 3 has a C, so 3's will bind there. On its side, 3 has a D. Let's see, D binds to B. Who has B? Number 2. So 2 is going to bind to the edges of the 3. Okay. The 2 interestingly has O's everywhere else, which doesn't bind to anything. So once the 2's are bound, that's the end. And so if you continue this process, what turns out is it doesn't matter what order you bring these blocks in, they're always going to form this little particular shape. And so that's an example, a very simple example of the kind of thing proteins need to do. They need to randomly float around and self-assemble into these very well-defined shapes. So now I can turn the question around and ask myself, if I have this particular shape that I want, can I find a set of rules, a set of blocks and rules that will make it? Well, I can do that by thinking hard about it. There's graph theoretical techniques, but it turns out I can also just do it by a kind of brute force evolutionary method. So I'll put down a set of random blocks and seeds. I'll see what they make, and a set of rules, and then I'll pick the ones that are closer to the shape, and then I'll re-randomize them. I'll mutate them. I'll keep doing that until I find something that gives the shape, and that works relatively well. And then my student Ian, who's now a professor at the University of Bergen, came up with a really brilliant idea. He said, I'm going to run an evolutionary simulation, and the only thing I'm going to tell it is I want to make 16 mers, so 16 little squares. It turns out that there are 13,779,255 different possible 16 mers. So you might think that if I told an evolutionary process make a 16 mer, but didn't tell it which one to make, that it'd make them all with the same probability. But what Ian discovered is something I think very exciting and very surprising, is that only 21 of these shapes appeared about 50% of the time. And that's, of course, for the same reason as we just saw for the RNA. They're the shapes that are easy, that have low complexity. In fact, if you plot the probability of getting one of these shapes versus its algorithmic complexity, you see this beautiful exponential decay. And there's another really fascinating thing about this, which is there something about these blue high frequency structure that's interesting? Well, there is. So dark blue here, I've given you 5 shapes. These have D4 symmetry, the symmetry of the squares. I can rotate them and flip them, and they're themselves. There are only 5 D4 symmetry 16 algorithms in the entire space of 13 million. And yet those 5 are going to appear with 35% probability if you randomly pick them by picking random rules, or by randomly evolving to them. They're going to appear very likely. So what's really fascinating is by randomly searching in the space of rules, evolution is going to find highly symmetric structures with exponentially higher probability than you might naively expect. And so we then looked at real proteins in the protein database. So we looked at a database of 34,000 protein clusters that a collaborator has made. And then we use the same complexity measure to see how complex they are. And we see that we can actually predict almost exactly like our polyaminos, the same probability complexity for real proteins. So this tells us that real proteins are also following this exponential algorithmic kind of coding theorem law. You can look at this in more detail. So I can look at, for example, 6 mers, right? There's a lot of 6 mers in the protein database, but this is a measure of their complexity, how complex, how hard they are to make, how many evolutionary steps you need to make them. And the high frequency ones are indeed simple, and the low frequency ones are complex. So this is kind of interesting. I've been able to predict for you how often self-assembling structures appear in nature. And what I see is with extremely strong bias, I'm biased towards structures with short descriptions. And of course, high symmetry is related to low algorithmic complexity. Because if something has high symmetry, it basically means I have a unit and I repeat that unit a few times. That's a low complexity description. Where something does not have symmetry, the only way of describing is by describing the entire shape. And so what we're what we're claiming here is that the fact that these protein clusters are highly symmetric, which is known, is not because nature has selected for them, but because highly symmetric ones are much more easy to evolve than non-symmetric ones. This is another nice example of something we see in nature, something that seems simple, something that seems beautiful, that naturally emerges from this algorithmic picture. And the last question I want to look at in the last maybe 7 minutes or 8 minutes that I have is another completely different application of this question. I'm going to look at deep learning. So deep learning, if you haven't heard about this, is a kind of technique which has revolutionized artificial intelligence. You know, big companies like Google and Facebook, Microsoft have poured many, many billions into this. Google DeepMind in London, very famous. They used deep neural networks to win at Go. They beat the world's greatest Go player, Lee Sedol, at Go. They just recently made a huge steps forward in protein folding. It's an absolutely extraordinary technology that will revolutionize our lives in various ways. And the fascinating thing about them is they work in a very simple way. So let's say I give a bunch of images, like these ones of cars and animals. Each image has a label with it. This one says car, this one says, you know, bird, for example. Then what I do is I take a whole bunch of ones that I know, and I feed into this neural network. And the way the neural network works is it's got an input layer, it's got a bunch of random weights on the inside, and then has an output. And I can then train it by giving it a whole bunch of these images where I know the answer, finding a set of weights that will give me the correct answer for this particular training set. And then if I give it a new image, it should, if it works well, recognize whether, you know, this is a bird or an animal, for example, or a bird or a car. And that's essentially how it works. Now, what's really surprising about these neural networks is the following, that you may not realize if you use them, but they typically have a very large number of parameters. So although I might feed it 10,000 images, it might have 10 million parameters. And so they're using what's called the over-parameterized regime. In fact, you can prove that neural networks are highly expressive. They can approximate any function. They're like universal function approximators. And all of kind of classical learning theory tells us that really, you know, having too many parameters is not a good idea. And in fact, physicists have taught us that for a long time ago. There's a very charming article by Freeman Dyson, who just recently passed away, about a visit he had to Enrico Fermi, where he had a 5-parameter model. He was trying to look at something, the weak interaction. And Fermi said, you know, my friend Johnny von Neumann said, with 4 parameters, I can fit an elephant. With 5, I can make him wiggle his trunk. And Fermi's point was, if you put so many parameters in, you know, you're not going to, you can fit anything, right? And you have no idea whether it's actually telling you anything about the truth. And so that's something we teach our students from a very young age. If we give them, if I give you, let's say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 data points, and you fit a 20-order polynomial to it, well, you'll be able to fit the data really well, but there's many ways of fitting a 20-order polynomial to 10 data points. And most of those will give you lots of wild oscillations that probably are not correct. Here's something interesting. Here I show you these 10 data points, and each of these solid curves are neural network fits to the data, different widths of different depths of neural network, but they all give you this very smooth curve. The fact is, the neural network can do any function, so any polynomial that you could fit to the data that neural network can represent. So it could do any kind of function. Why does it pick this particular function? That's kind of strange, isn't it? And so that's a question that we've got interested in. And I'll give you another example of how this might work. So here's a very simple learning question. Let's say I am going to try to teach some kind of machine learning algorithm to predict whether it should send you to the hospital or not if you come with a certain number of questions. So you come to a doctor, you say, he says, do you have a fever? Yes, that's a 1. No is a 0. Cough, yes. You've lost your sense of smell. You're older. You've got heart problems. You're obese. You have diabetes. Then the doctor might send you to the hospital because you have a, you know, that looks perhaps worrying. If you have all those symptoms, but you're young, but you've got, you know, various kinds of comorbidities, you might send you to the hospital too. If you've got the symptoms, but you're young and otherwise healthy, they may say, well, you know, sorry, mate, stick it out at home, etc. And so I have a very good doctor. I could go through those questions. And now there's 7 of these questions. So there's 2 to the 7 possible answers that you could give to those questions. That's 128 answers. But for each of the answers, I could have a different response, send you to the hospital or not. So there's actually 2 to the 128. There's actually, in a simple problem already, 3 times 10 to the 38 possible outcomes. So what you could do is you could give your machine learning the first, say, 64 answers and wonder whether it could give you the correct answers on the other 64 possible ways of answering the question. And so the way to think about this is what's really happening in these neural networks is you've had a bunch of parameters that you're trying to fit. So I might give you 64 of these answers and ask the machine to predict the other 64 and see how well it does. I do that by basically fitting these parameters. And what this is basically doing is it's taking a function, because the function is taking all those 128 possible answers and fitting to them. And so the question is basically these neural networks are input-output maps from parameters to functions. And that's really interesting because we've just looked at input-output maps. And so we predict that these should have simplicity bias, and indeed they do. So if you randomly pick parameters, that's like randomly typing on a typewriter, randomly picking sequences, then we're predicting that you should be exponentially more likely to get simple functions rather than complex functions. So we have a measure of the complexity of the functions that are given. So the simplest function in this case is just send everybody to the hospital or send nobody to the hospital. That's the simplest function. And that one's the one that appears right here with about a 10% probability if you look at a neural network and just randomly pick parameters. So the simplest function is the most likely function. And then as you go down, other functions become less and less likely. Remember that there are 10 to the 38 possible functions. So roughly with 1 in 10 to the minus 38 is the mean probability of a function. Yeah, all these functions here have much higher probabilities. And so these neural networks are very highly biased towards these simple functions. And the last thing I might tell you about this is this helpful or not? So neural networks are biased towards simple functions. Is that going to be good? Well, it's only good if if the doctors are giving you simple answers. So those are answers that have some kind of structure to them. And so here I show you the error that the neural network makes if I train it on half of the answers and ask how well it does in the other half. For simple targets, so for doctors that are giving you relatively ordered answers, to very complex ones, doctors are giving rather disordered answers. So what you can see if you look at the questions I did before is there's a structure to them, right? So young people who don't have comorbidities, doesn't matter what their symptoms are, they're not going to be sent to the hospital, for example. So those are the kind of structure in there. And so that structure basically tells you there's simpler ways of describing the data than just looking at all of it randomly. And so this neural network does extremely well. And it does much better than what's called a random learner. A random learner would be the one that picks a function that gives you 0 error on the training sets, but doesn't look at anything else. There are actually 2 to the 60. If I give you 64 of the 128 questions, there are still 2 to the 64 functions that will give you 0 error on those 64 questions and predict something else on the other 64 outcomes. So there's many possible functions. Pick one at random that you've got 2 to 64 functions you could pick from. Almost every one of those functions is really bad. But neural network, because it's biased towards the simple functions, is picking functions that look for that are simple. And since most of the things that we're interested in nature and in science and in engineering have some kind of structure to them, that bias towards simplicity, that kind of Occam's razor-like bias, is extremely important for making these neural networks work really well. So we're basically claiming that neural networks work well because they're biased in this very powerful way towards simple functions. So I told you 3 stories in hyper-astronomically large spaces. Think of Borges's library. The possibility spaces are huge, but simpler stories are exponentially more likely to appear than complex ones. Looked at RNA, I looked at protein, I looked at RNA folding, I looked at protein cluster self-assembly, and I looked at machine learning. And as always, the people that did all the work are listed here. So Ian, Stefan, Kamal, Chico, Nora, Guillermo, Chris, Vaibhav, and Joachim are some of the main protagonists in this story. And I'm very lucky that I've had such an extremely talented set of students to work on this. I'm just the one that goes around and tells the stories. So thank you very much for your attention, and I look forward to listening to your questions.
Thank you, Ard. That was really, really interesting. It's a fantastic way for us to kick off our evening talk series. So as you've given us 3 stories, I think it's quite nice that we've got 3 really interesting questions, I think, to ask you. So based on the first third of your talk, Penny's posed a question which was, does essentially then nature ignore the most complicated possibilities? And I think we're referring to the protein fold here, because they would require simply too much energy. Is it, can we simplify it to that level, or is that a potentially an oversimplification?
Yeah, so that's an interesting point. So I'm not saying anything about energy really, or even about free energy. I've simply said in these kinds of problems where there are very large search spaces, then you're much more likely to find solutions that are somehow simple. And that's because there are many more ways of making a simple solution than there are ways of making a complex solution. Is basically the story. So just like in Borges' library, if my story is effectively simple, I can have many books will tell that story. And so I randomly pick a book, I'm quite likely to find a simple story. But a very complicated story that needs all 410 pages to be told really can only be encoded in one way, which is that particular book, because it's going to be unlikely to be found. And that's the story. So it has nothing to do with energy or even free energy, although I use free energy minimization to find what the structure is that a particular sequence gives. But we didn't find any correlation between those free energies and the likelihood that they appear upon random sampling. So this is basically this idea that simple things are more likely to appear than complex things comes from the coding theorem that was proved by Levin. And I think it's sufficiently general and interesting that it ought to be taught in our undergraduate curriculum. I don't understand it well enough yet to be able to teach it, but I'm hoping that I will one day.
Fantastic, thank you, Ard. And actually, I think you began answering the second question, but I think it's still worth posing because it's clearly something that's cropped up in the chat on more than one occasion. So Tariq has very kindly asked, does this discussion therefore prompt us to think of energy as a factor that breaks symmetry? The universe is not quite symmetric, isotropic because of energy, question, which makes us ask again, what really is energy and whether energy itself is isotropic in the universe? Is there anything you'd like to add to that?
Yeah, that's a very deep and interesting question. I don't quite, so in order to answer questions like that, you've got to have some kind of theory about how universes are generated. And I think, although there are lots of speculative ideas about that, there's not really anything that's well enough understood or likely enough to be true that we can get very far with that. But I mean, if you, yeah, who knows? There was actually interesting in just this month in Aeon, AEON, which is an online magazine. So John Joe McFadden, who's in Southampton, I think it is, Sussex, University of Sussex, who's a popular writer, has an article about simplicity and universes, where he basically says perhaps there are many universes, and somehow simple universes are more likely to generate life than complex ones. Who knows? I think this is pretty much rank speculation, but there's nothing wrong with speculation. Sometimes it might cash out. So quite fun to think about.
Superb. And we've actually got two final questions, which has broken my own symmetry there for wanting three. But nonetheless, Kushi's asked us, clearly the inverse relationship between complexity and probability has been empirically established. What is your view on whether what does this mean essentially for multiverse theory? Now that might be somewhat out of your field, Ard, but I'm going to post it to you nonetheless.
Yeah. So okay. So the so I don't know what it means for multiverse theory, because multiverse theories are not very well established. So I have so there's different ways of making multiverses. One of the ways is that you look at very high dimensional structures in string theories, like Calabi-Yau manifolds, that have many possible solutions to them. Again, something like 10 to the 500 solutions would be one way of thinking about it. We call this the landscape. And so what you do if you're a string theorist is you try to look for what's called compactification. So you try to, these are in high dimensions, so you try to roll up the higher dimensions to some kind of lower order theory that we might understand better and see what those look like. And so it would be very interesting to see whether that would give some bias for simplicity or not. I've talked to my colleagues off and on about it, but they kind of roll their eyes and say this is going to be pretty hard. I mean, the real point is, here's an interesting question about multiverses, which is, what are we doing as physicists when we start thinking about multiverses, things that are probably never going to be empirically verified. Are we still doing physics, or is this really some kind of applied philosophy? Right? We don't really have, I mean, I'm a theoretical physicist, so I'm a big believer in theory, but we know from experience that that experiments are incredibly important in science in helping us move forward. And it's very easy that we also have experience that when we speculate without any experimental income, any kind of experimental input, we can easily get led astray. So I think there's a very interesting question here about multiverses, which is a kind of meta-philosophical question, which is, what are we doing as scientists? Have we moved into some kind of philosophy? Should we therefore use some of the modes of interrogation that philosophers have used over time, or are we doing actually just science, but a different kind of science? And I don't really know. But I have, back to that question, it's a very nice question, because I have actually thought about some of these theories that might be linked to multiverses and wondered whether there might be a bias towards simplicity in them. Of course, I'm biased towards simplicity bias, yeah? So I think there might be one, but until I calculate it, I'm going to have to keep an open mind.
Fantastic. And hopefully Matt's still there. Matt posted in 2 questions, so I'm going to hand over to Matt, one of our attendees, to ask the final double part question, which starts with a question regarding external factors affecting simplicity of folding RNA. So Matt, if you're there, if you'd like to ask your question, that would be great.
I am. Dr. Louis, do you think there are any external factors that affect the simplicity of folding RNA? And I've used solar radiation as an example, but this can be anything such as the chemical makeup of the atmosphere or anything.
Well, I think the answer is yes. So I should be very careful. So I've only looked at secondary structure, which is who binds to whom. The question of predicting the tertiary structure, so exactly what the 3-dimensional structure is very hard. And one of the reasons why is that the salt, for example, does really strange things to RNAs. And we know that if you take an RNA that folds well in the cell and you put it in your test tube and you don't get the salt right, it won't fold, or it'll fold in something different. So they are quite sensitive to this. And you can think of the secondary structure as a slightly coarser grained way of looking at them, that picks up some coarser features, but might not get all the fine-grained features correctly. So RNA is an interesting molecule. On the one hand, it's incredibly fickle and difficult to work with, which is why you'll find physicists there's like a lot of physicists that work on DNA. I do a lot of predicting of DNA shapes, and I've got colleagues here in physics that work with it as well, because DNA is a relatively easy molecule. And we always say to each other, we should work on RNA because RNA is more chemically interesting because it can do all these. In our bodies, DNA doesn't really do anything. RNA does tons of stuff, so we should use RNA. Where, all my experiments says, RNA, you put it in your test tube and it degrades, and all kinds of terrible things happen to it. And this is interesting. The reason I am banging on this, I think this is what I'm riffing on your question, which is that one of the biggest ideas for the origin of life, so where did life come from, is RNA, because life needs at least 2 things. It needs replication, so you need to copy yourself from generation to generation with some fidelity, and you need metabolism. You need to be able to take energy from other sources and use it, so you can make more of yourself. So the problem is that DNA does replication, but doesn't do any metabolism. Proteins do metabolism, but they can't do replication. So you've got a chicken and egg problem. And so when people realized that RNA can both encode information and can do catalysis, so can do metabolism, it was obvious to say, well, maybe life started as some kind of RNA life. And it's a very charming idea, and there's some indirect evidence that this might be true. So some of the very fundamental properties of the cell, like the ribosome, have a lot of RNA in them, which suggests maybe they had an RNA precursor. But of course, the problem that a lot of my colleagues tell me is that you'll try working with RNA. It's an incredibly fickle, messy molecule. The solar radiation, as you point out, affects it. The salt affects everything, affects it. It breaks down all the time. They think there's no way that on the primitive Earth it would have ever survived. So who knows, right? That's an interesting question. Maybe that the very first life was based on very simple RNAs, for whom the folding was much more robust, and then eventually more complex ones evolved. Would be one of the ideas. Maybe something completely different happens. The real answer is we have absolutely no idea how life originated, and that's fine.
And the second part of the question, which is, it's not relevant to my current level of study. I'm only a first year physics with astrophysics student. Is there any resources where I could view and learn a little bit more about this RNA, folding RNA? Because you've actually piqued my interest in this.
Yeah, no. So if you type it into Google, there's actually quite a bit on it, and the reason there is because there's a bunch of people that have programs that you can. So a good type in Vienna RNA, because one of the most used programs is from the University of Vienna. It's the one that we use. And if you go on there, you'll find a program you can literally download onto your laptop, and you can start folding. In fact, even better, they've got online ones where you can put a sequence in, it'll fold it for you. You can start playing with it, almost like a game. It's quite fun to do, and that's a good way of getting some intuition.
Fantastic.
So Vienna RNA is a good place to start.
Thank you very much for the talk today. It was fantastic. Thank you.
Thank you for your excellent questions.
Brilliant. Well, that actually brings us nicely to the end of our proceedings. So firstly, from myself and everybody at the Keele IOP Centre, thank you very much, Ard, for a fantastic talk. It was on a subject we have not covered at all as a centre for a long time, so it's been really fascinating. Thank you to everybody that's been listening. There's been some fantastic questions. And just before you go, just to remind you that our next talk is being given on the 18th of November by Dr. Juliana Moorbeck of the University of Keele on the subject of two-dimensional materials. So thank you very much for listening. I hope you have a lovely evening wherever you are, and all the best. And I'll see you next month. Thank you very much.
Hi, Scott. Just a quick question. I can see there's a transcript of it has been made of the talk.